Hello, can everybody hear me?
Yes, we can hear you.
Yes.
All right, my computer is going to be really slow and overworked because it's processing the lecture file. So please bear with me. Let me see what I can shut down. I can shut that down. And I can probably shut that down. Let me just talk briefly about the syllabus. We're going to be covering Lab 7, throat and urine cultures. And if possible, I'm going to try and send you your results for the first 3 tests, which I chose. The problem is the files that I want to send you are on the computer that crashed. And I just heard today that they cannot get into the hard drive and give me the files. So I've got to remake the files. So I might be a little late on getting you your results. All right, any questions about any of that? I will try and get it today, but I've got to remake the file and I've got to remake actually 3 files. All right, let's close that down and let's start sharing my screen. And for some reason, this version of Zoom on this computer is not showing anything. It should show me in a background, and I'm not sure what the problem is. All right, let's go to the share screen. Am I sharing my screen? No. There we go. I clicked on it and nothing happened. Oh, that's telling you the meeting from the lecture is converting. It's only at 45% now. It'll probably finish before I finish talking. All right, Lab 7. Hopefully this is a new version of Lab 7. You do have some reading to do before you start Lab 7, so take a look at that. It's not too much. You need to read about the nose, the throat, and the mouth on page 392. You should read about the beta hemolytic streptococci, and that you can see the text at the end of this lab module. You should read about Haemophilus, that is also in the text at the end of this lab module. You should read about the oxidase test, and that you can get from page 132 of your textbook, and the catalase test, page 155, and the coagulase test. There's some text at the end of this lab module. So the learning objectives for today: upon successful completion, you should be able to explain how to properly collect throat and urine specimens for routine culture. Be able to list the common bacterial pathogens isolated from the throat and urine cultures. Know the different culture medias we use to cultivate these pathogens. Recognize which tests are used to identify throat and urine pathogens. And then lastly, understand the principles and how to interpret the following tests: the catalase test, the coagulase test, and the oxidase test. We will talk about the XV factor testing, but you don't need to really understand it or interpret it. I mean, I'm not going to test you on it. Once specimens are received in a lab, they are logged and processed in a timely fashion. The specimen will be grown on specific culture media that are inoculated and incubated for that specimen. We're going to talk only about throat and urine specimens today. Below is an overview of the media that are used to cultivate common bacterial pathogens isolated from the throat and urine cultures. For the throat, we really only have 2 culture media that are common for throat pathogens. And blood agar, specifically for Streptococcus pyogenes. And then the second is chocolate agar, which is for Haemophilus influenzae, which is primarily seen or mainly seen in small children and infants. For urine specimens, we have 3 different media we use: blood agar, and then one of the other 2, McConkey's agar or eosine methylene blue. And McConkey's agar is abbreviated MAC, and eosine methylene blue is abbreviated EMB. For these, we look for the pathogens Streptococcus, Enterococcus, Staphylococcus, and species of Enterobacteriaceae, largely E. coli and other enterics, as well as Pseudomonas. Now, for the Enterobacteriaceae and the Pseudomonas, we will grow them on McConkey's and EMB, because these plates are selective for gram-negative, and they're gram-negative. For the other species, we grow them on blood agar, not because they require blood agar to grow, like obviously Staphylococcus species can grow on general purpose media, but it can grow in blood agar as well. But then there's Streptococcus, which at least most species do not grow on general purpose media, so we grow it on blood agar. And blood agar will also grow Enterobacteriaceae and Pseudomonaceae, meaning the Pseudomonas. It's not selective. For throat cultures, when we're talking about them, you should realize that sore throats can be caused by viruses as well as bacteria. Usually a doctor can look in the throat and determine if it's a bacterial infection. If they see white patches in the sore throat, maybe on the tongue as well, that's a bacterial infection. If the throat is just red, that's a viral infection. The most common bacterial pathogen, meaning a bacteria that's causing the sore throat, is Streptococcus pyogenes. It has a couple of other names. I will always call it Streptococcus pyogenes, but it's also called Group A Streptococcus or Group A strep, and occasionally GAS for an abbreviation. For a patient who has a strep throat, it's diagnosed in one of 2 ways. There's the rapid test or the throat culturing method. We're going to talk about the throat culturing method, where the clinician swabs the back of the throat and the tonsils where there's redness and white patches, and they do not touch the tongue or the sides of the mouth. And usually you go in with a, what do you call that? Tongue suppressor, as well as a swab. Patients that test positive for Streptococcus pyogenes are placed on antibiotic therapy. You can read about that, but generally Streptococcus pyogenes is not fought well by our own immune system. So we place the patient on antibiotics. Meaning most patients cannot throw off Streptococcus pyogenes infection. Streptococcus pyogenes is fastidious, and so it is grown on a culture media supplemented with 5% blood, or we call this blood agar plate, or BAP for an abbreviation. The plate is then examined for any small, clear colonies surrounded by complete zones of hemolysis. This is beta hemolysis. And if it has a beta hemolytic colonies, which are small, then it could be Streptococcus pyogenes. But there are a few other Streptococcus which have a similar beta hemolysis. So we do 2 biochemical tests to show that it's Streptococcus pyogenes. You run the catalase test and the bacitracin susceptibility test. So if you have, let me see if I can blow this up. This is bacteria growing on the plate, and that looks like alpha hemolysis because it's darkening. This right here, well, maybe not. Let me blow that up further. That might be gamma hemolysis right there, but that might be very small alpha hemolysis, very slight. This is the growth right there, if you can see that, and right there. This doesn't have any hemolysis around it, but these do, the white regions, that's beta hemolysis. So if we get that, we then run the catalase test. Shrink that down now. In the catalase test, if the bacteria have catalase, we then expose the bacteria to hydrogen peroxide, and then the catalase will decompose the hydrogen peroxide, producing water and oxygen. The oxygen would create bubbles in the hydrogen peroxide, so you see the bubbles. Most organisms that grow in the air will have catalase, and they will be positive for the catalase test. This is positive where you can see the bubbles. This is negative. However, the Streptococcus species, as well as some Enterococcus species, test negative for catalase production. So if it's positive, it's not Streptococcus. If it's negative, it might be Streptococcus, and it might be Streptococcus pyogenes. You then run the bacitracin susceptibility test. Bacitracin is an antibiotic, and it's in the disc here. You take the bacteria from the blood agar plate, that is beta hemolytic, and then you smear it all over the plate. And if the bacteria are sensitive to bacitracin, it is Streptococcus pyogenes, called group A strep here. If the bacteria are resistant to bacitracin, it is not Streptococcus pyogenes. It's some other species of Streptococcus. Any questions about any of that? All right. Another organism that can be clinically significant from the throat is Haemophilus influenzae. It's a gram-negative coccobacillus. It is a common cause of ear, eye, and sinus infections in babies and young children. Fortunately, older children and adults typically do not get infection with Haemophilus influenzae. Haemophilus influenzae is another fastidious organism. However, it does not lyse the blood in a blood agar plate. And so that's why we grow it on chocolate agar. Chocolate agar has the red blood cells lysed. That's what gives it the brown color. And Haemophilus influenzae will grow on not blood agar, chocolate agar. To further prove Haemophilus influenzae, you don't need to know this. You can then run the the V factor test, looking for NAD, and the X factor test. And like I said, you don't need to know that. Curiously, Haemophilus influenzae, it won't grow on blood agar, but if you grow it on blood agar, it also has Staphylococcus aureus on the plate. Like here, we have a single streak of Staphylococcus aureus coming along here, and that's that white streak this way. And then we streak the plate this way with Haemophilus influenzae. You can see that in this region of the plate, no Haemophilus influenzae is growing, but in this region of the plate, near the Staphylococcus aureus, Haemophilus influenzae is growing right here and right there, all along there. The reason is that the Staphylococcus aureus is lysing the blood in this region, and the growth factors from the blood cells are then diffusing out to this region, and that the Haemophilus influenzae can then use those growth factors and can grow in this region. But beyond this region, the Haemophilus influenzae do not grow. Any questions about any of that? Let me state that Haemophilus influenzae was misnamed by Louis Pasteur, and he thought it caused the flu because he got it from a flu patient, but in reality, a virus causes the flu, not Haemophilus influenzae. And in Louis Pasteur's defense, he isolated Haemophilus influenzae before Dr. Koch wrote the Koch's postulates. So Haemophilus influenzae is misnamed. It does not cause the flu. Any questions about any of that? Any questions about throat cultures? If not, let's move on to urine cultures. Urine cultures are used to diagnose urinary tract infections or UTIs. The bacteria can travel from the urethra up to the bladder. Females are more predisposed to developing UTIs than males simply because they have a shorter urethra. There are different ways to collect a urine specimen. If you're using, and let me show you the three ways, a clean catch midstream, catheterization, and suprapubic aspiration. In clean catch midstream, you clean the genital area before the patient voids the urine, and then you catch the urine for the specimen in the middle portion of the urine, and that's to get it cleaner, because when you're urinating, it goes through the urethra, which has many bacteria in it. And so we do a clean catch midstream just to reduce the bacteria that you get from the urethra. An even better way of getting a urine specimen is to put a catheter into the patient's bladder and then sample the bladder directly. This is a somewhat invasive procedure, especially for a male, but it is cleaner. The trouble is the catheter is put through the urethra, which does have bacteria on it, and that can put bacteria into the bladder. So it's not perfect either, it's just better. And then the best way, which patients really hate, is where you have suprapubic aspiration. You put a needle through the patient's abdomen into their bladder and then sample
Do Botox that way.
bladder directly. Somebody, you have your, thank you. You have your microphone on. This way is extremely clean, but patients don't like it because obviously you're sticking them with the needle and sticking it into the bladder. Those are the different ways to collect urine. Once the urine is obtained by the lab, you then put 0.001 milliliters. That's 1/1000th of a milliliter of urine on a blood agar plate. We have a special inoculating loop that measures exactly 0.001 milliliters, or 1/1000th of a milliliter. That's actually 1 microliter. Let me show you that. Well, I thought I had a picture of that. Apparently on this version, I don't have a picture of the, oh, there it is. I did have it. It's right there. So that's the loop you use to put in 0.001 milliliters. And then you streak it like that on the blood agar plate, and then like that. In addition, for urine samples, besides growing the sample on a blood agar plate, you also inoculate either a McConkey's plate or an eosine methylene blue plate. And this will grow only gram-negative bacilli, McConkey's and eosine methylene blue. Why you want to put it on a McConkey's or eosine methylene blue plate is the most common urinary tract infection of a bacteria is E. coli. And other coliforms also are common. Okay. And when we put in all of the enterics, and maybe the Pseudomonas as well, it's over 50% of the species. Okay. Anyways, E. coli is the most commonly isolated bacteria causing a urinary tract infection. So we grow it on mock as well. The interpretation for a positive urine culture is multifaceted. There are different factors to consider.
Touch, just look and listen.
Can people turn off microphones? I have it set, I thought, so that your microphone was initially turned off. I can't get anyone. So hopefully we won't have trouble again. One thing is the type of collection, whether it's the clean catch voided system or whether it's the catheterized specimen or the the puncture into the bladder. The gender of the patient also determines the interpretation. I think females have more contamination. I'm not sure why that is the case, but they have more contamination of the urine specimen. And then the number of colonies seen per mil of urine also determines the interpretation. The presence of polymicrobial growth affects the interpretation. If there's many different types of organism, it probably was a poor specimen collection. But if there's few polymicrobial growth, meaning it's mostly one type of organism, it's probably a better collection of the urine sample. Yeah, see here, males get less cells per mil than females. I don't know why that is, because you'd think the males have a longer urethra, that they would have more. All right, where do I scroll down on this? Lost my mouse. There it is. All right, so once you get growth, you then start doing some tests. One test that you can run is the coagulase test, and it allows you to distinguish Staphylococcus aureus from other gram-positive bacteria, including other members of the species Staphylococcus. So if there's production of the enzyme coagulase, that means it's that the bacteria causing the urinary tract infection is Staphylococcus aureus. And if it's a coagulase negative, then it's either some other Staphylococcus species or some other species. So coagulase is mainly used to distinguish Staphylococcus aureus from the other members of the family Staphylococcus. What happens is, in the coagulase test, you put fibrinogen in with your bacteria. If the bacteria produce the enzyme coagulase, it starts creating a clot. What happens is the fibrinogen is changed into fibrin. What that looks like is on a slide, you'll see clumping, negative, coagulase negative. And on the test tube test, the coagulase test will be liquid. And if there's clumping, coagulase positive, you get clumping or clotting. Clotting would be a better word. Any questions about any of that? If you see any clot formation or clumping, then it's coagulase positive. All right, E. coli is the most commonly found bacteria from a urinary tract infection. E. coli is the family Enterobacteriaceae. All the members of this family can ferment glucose. So if it ferments glucose and it's gram negative, it's probably Enterobacteriaceae. And that's one way we can distinguish between Enterobacteriaceae from the family Pseudomonaceae. Another way is to run the oxidase test, and the Enterobacteriaceae will be oxidase negative. Pseudomonas, especially Pseudomonas aeruginosae, or aeruginosa, I'm having trouble saying that now, is another gram-negative bacillus, but it doesn't ferment glucose, and it's an aerobe, Enterobacteriaceae, or facultative anaerobes. And then the Pseudomonaceae are oxidase positive. So you can run the oxidase test. Oxidase means that cytochrome C is present. Pseudomonas aeruginosa is oxidase positive. And if it's oxidase positive, the oxidase test will turn blue within 30 seconds, or you could say purple, bluish or purple. After 30 seconds, you should not rely on the test, because the test itself may turn blue or purple after 30 seconds, even if the specimen is negative. So you should make your reading from 20 to 30 seconds. All right, any questions about the urine specimen? All right, you can read about the summary, you can skip the references. I think I've got this in the exercises, but let me check. Yes. So in the laboratory 7 exercises, you're not going to be performing any of these activities. However, you will be held responsible for knowing how to perform them. There are some slight procedural variations for how you perform each test, but we will go with the video clips that we're showing you. And there's a video clip for the catalase test, a video clip for the coagulase test, and a video clip for the oxidase test. So watch the different video clips. Make sure I've got everything here. And then answer the questions. So identify the principle used to detect the presence of catalase, coagulase, and oxidase. What converts fibrinogen into fibrin? Name an organism that tests positive for this enzyme. And so there'll be a 1A and a 1B. And 2, what detects the presence of cytochrome C proteins in the electron transport system? It's one of the tests. Name 2 organisms that test positive for this enzyme, and they're mentioned in the clip. 3, what hydrolyzes hydrogen peroxide into non-toxic products, water and oxygen? Name 2 organisms that test negative for this enzyme. And obviously Streptococcus pyogenes is one. 4, what rapid test is used to differentiate between the following? How can we rapidly distinguish Staphylococcus from Streptococcus? How can we rapidly distinguish Staphylococcus aureus from other members of Staphylococcus? And how can we distinguish the family Enterobacteriaceae, such as E. coli, from the family Pseudomonaceae, such as Pseudomonas aeruginosa? And then 5, concerning specimen collection for a routine throat culture, what areas of the throat should be swabbed? What areas of the mouth should be avoided? What is the most common bacterial cause of pharyngitis? That's a sore throat, and you must state a bacterial cause, because obviously the most would be all viruses, but we're talking about what's the most common bacterial cause of a sore throat. What test is used by microbiologists to differentiate between beta hemolytic I think this test is supposed to be skipped, but that's not crossed out. Let me take a look here, because I don't think we talked about that. I don't have it crossed out here either. Oh, I'm sorry. We do discuss that. So you do need to answer 7. It's 8C that's crossed out. 8, answer the following concerning a child's throat culture. State 2 non-selective agar plates that are inoculated for a child's throat culture. And what organism can be isolated on CAP, but not isolated on BAP? And 8C you can skip. That's why it's crossed out. 9, list 3 methods that are used to collect urine for routine culture. We did discuss them in this lesson, actually. List 3 reasons why urine specimens should be rejected for culture. So if somebody collects the specimen inappropriately, that would obviously be one reason, and you can state specifically what that was. How much urine is placed onto the surface of a blood agar plate? And what is the most common bacteria isolated from patients with I'm not saying this right, cystitis, and that's a urinary tract infection. And then 13, identify the Gram reaction and the cell morphology, the shape of these bacteria. You may need to look these up. Most of them are discussed in the lab or in the handout to the lab module, meaning there's some text after the lab module. I'm going to scroll to it now. The links in this do not work, but the text is correct, okay? So there's some information after the lab module. So what's the gram stain of the Staphylococcus saprophyticus and its cell morphology? What's the gram stain of E. coli and its cell morphology? The same for Pseudomonas aeruginosa, Streptococcus pyogenes, and Haemophilus influenzae. Any question on any of that? All right, that's it. I will be here until 8 o'clock to answer questions. This lab is actually not that difficult. The next couple of labs will take you much more time to do than this one, but this one, everyone should be done probably by 8 o'clock if they work on it now. I will be here to answer questions, although I'm going to go get my dinner, so I might be away from my desk for a few minutes. If I don't answer you back, just try again a few minutes later. Unfortunately, you can't see me, so you won't be able to tell if I'm here or not, but just talk to me. I'll keep the microphone on. Any questions? All right, if not, lab, and I'm going to stop recording.